All right, good morning, everyone. Let's finish off cycle 11 on epigenetics. This is part 2. This is your last lecture for this course. So please take the remainder of the time to do whatever you have to do to catch up or to study and to really be prepared for that final exam and hit a home run. Before we start lecture, just a few announcements. The final exam tab is now available on Brightspace. So please go to that tab and read all the details carefully, all the policies, all the expectations, so that you can arrive fully prepared. The exam takes place April 15th from 9 a.m. to 12 p.m. It's a 3-hour exam. There are 45 multiple choice questions and 5 short answer questions. In preparation for your exam, we have a few things available to you. First is that we will post a practice final exam on that tab. Please use that practice just to get an understanding of just some example questions. Dr. Maxwell and I will host a Q&A session, and we will post that detail. We'll also send an announcement. The Biology Mentorship Program will also be hosting final exam review session that you can attend. Hopefully you'll find that very helpful. They're a great group to help you study for the final exam, so please take advantage of that. Since this is the last lecture, I won't have any opportunity to make announcements in class and post the lectures with the announcements. So please make sure that you look out for announcements on Brightspace. It also comes to your email address. It's really important because this is the only form of communication we will have left. Anything can happen, things can change. We could have an emergency and we have to pivot and change things for the final of the course. It's really your responsibility, please, to make sure that you are on top of and cognizant of the fact that an announcement could be coming and it could have important information. It's your responsibility to read the announcement and be aware of any changes that may or may not come. Exam day reminders, please clearly write your name, username, student number, your name as it appears at the registrar's office. Please bubble the correct version and all the answers completely. If you make erasure marks, then make sure that you erase completely and fully if you're changing your answer. Any incomplete or incorrect bubbling is going to affect your grade, unfortunately. I'm not sure if you guys are aware, but since September, there are many students that are not writing their name correctly, their username, especially the bubbling. They forget to bubble their version. For their answers, they're not erasing properly. And the system comes back and tells us things like there are 689 questions that have 2 answers. Should I go ahead and give them all 0? All I have to do is click one button and say yes. But we don't do that. You know what we did? We went manually with our eyes through all of them and said, oh, look, this student actually put B because A was not erased properly, but I can see with my eyes that are more sensitive that there was some erasing on the A. So we're going to give them B because they meant to erase A instead of a 0. So there was that. And then there were people that circled 2 answers, but then put an X through one of them instead of erasing it. So the system sees 2 dark spots. They're going to give them 0. But they double check to make sure. By the way, we have 279 questions that have 2 options circled with one of them, so when I go and look at it manually with my eyes one by one and I click manually what I want, I can see the X. I'm like, well, I think they meant to say, no, I don't want this answer, I want this answer. But please, guys, you need to erase and erase fully. We don't have the resources to do that. It's really tough. If you don't circle your version, the only way we can find out what version you wrote is to go through boxes and boxes of over 2,300 papers to find yours and then see what version you wrote on the exam paper. So that takes a lot of manpower to do that. And then we circle the correct version for you, scan it again, and then we grade it so that you actually have a grade as opposed to 0. So please be mindful, guys. This is the final exam. You really don't want to lose marks and have your GPA affected because of something like this. So please, I'm begging you, do it for me. Just think of all the challenges that come our way during grading. Office hours, I am going to be hosting office hours this week, which is today, from 2 to 3:30 in NCB 340. I will also be hosting office hours April 2nd and 9th. I don't have to do that. We don't have to host office hours the weeks that we're not teaching, but I think it would really benefit you if there are office hours available. Please come and ask any question you have. It doesn't have to be about a cycle. It could be just, general studying strategy. I'm stressed about the final. What do I do? Just a pep talk, whatever you need. Whatever you need, this is the time. Take advantage, because then after that, it's hard to get a hold of faculty. So, skill development survey. Kindly note that April 10th at 11 p.m. is a deadline to complete the survey. If you complete the entire survey, I will add a 0.5% bonus to your overall course grade at the end of the term. Unfortunately, if you do the survey after this deadline, I will not be able to apply the credit. Right? So please make sure that you complete that by April 10th. I would really appreciate that. I appreciate your feedback. I appreciate the time you're putting into it. And I hope the reward is something that's worth something to you. I can't give a huge reward for one survey. That would not be acceptable from an academic standpoint. So this is honestly one of the highest percentages I can give for a short survey. So today we'll be talking about how maternal care has an effect on the epigenome. This will be one of the topics. We'll also be talking about how changes in the epigenome can lead to diseases and conditions and syndromes, and what are some of the ways in which they're developing drugs to target the epigenome to fix the disease, to better treat the disease. So the epigenome is now becoming a target for treatment because the epigenome plays a role in causing the disease. So why not try an epigenetic drug? And they've shown the epigenetic drug working in many animal models, and there are clinical trials in humans that are using epigenetic drugs. So we talked about last class that twins, although they have an identical genetic sequence, they have changes in their epigenome, and you can see that change very early on. And so as you go through life and you accumulate mutations, that's genomic, and that's not very much. But your epigenome changes more drastically, or it has more significant changes. And as you age, you modify your epigenome along the way, and that is known as epigenetic drift. These are epigenetic modifications that naturally occur with age. So you can see here a scan of 3-year-old twins. They're identical monozygotic twins, and the scan of 50-year-old monozygotic twins. The yellow represents regions that are similar in methylation pattern. So if you look at the methylation in one twin and the other twin in that genomic region, then it's the same. This is just an example of shown here, but all chromosomes have been shown and they all have the same pattern. This is the pattern of 50-year-old identical twins. They're mainly green and red. So what are the green and red representing? The red regions are hypomethylation regions, and saying that one twin has hypomethylated region compared to the other. So one is more than the other. So the red region here is hypomethylated compared to the other. The green region says that one region is hypermethylated compared to the other. So look at this difference. There's a significant difference, mainly red and green, by the age of 50, but very little, but you can still see changes happening. And as I had mentioned, you can see these changes sometimes even in utero. So there are many topics that you can discuss with respect to epigenetics. We're just going to cover a few of them because of the time constraints. But this is something that really gained a lot of traction towards understanding the role of the epigenome in how we perceive the environment and the role of the environment, I should say, in the epigenome. This was the paper. So this was a landmark paper published in Nature Neuroscience. It's a very, very high impact journal. Many scientists go through their entire career and they never publish in Nature Neuroscience. This was called the Weaver paper in 2004. And this is a landmark paper, meaning that they discovered something and then this was reproducible in many labs across the world. And now here we are 20 years later, and a lot of studies have stemmed from this, strong supports for this. So that's why it's called a landmark paper. Landmark paper is not a paper where they discover something and they go, oh, well, we discovered it. Did you discover it? Did you find the same thing? No, we didn't. We couldn't reproduce it. Well, I don't know why. That's just a paper then. But a landmark paper holds a pretty big, heavy title. So what they discovered here, and this is the paper that convinced a lot of those scientists that were skeptical about behavioral epigenetics. Like, really? So as the stimulus is taking place, as you're experiencing something, your epigenome is changing, like methylation is occurring and demethylation is occurring as you're experiencing the stimulus? Yes, that's happening. And they proved it. They use rats. They use rat models. And this has been proven in mice, other animals, including monkeys. So what they did was they took female rats, or moms that were pregnant, and they gave birth. Upon birth, they noticed that some rats were highly nurturing rats, and some were not nurturing. Nurturing, in this case here, is through licking and grooming. This is how rats and mice nurture their pups. They lick and they groom. And that is their way of showing affection. So both groups received milk, they received all the comfort that they needed, physical comfort that they needed, right? Warm place to stay, good night's sleep, they had whatever they needed. But this mouse here, or this rat here, did not groom and lick, but this one did. So what the scientists noticed is they said, oh, I wonder if these pups, when they're a little bit older, I wonder if they behave differently than these ones. And they sure did. These pups were highly anxious. These ones were not. So you're like, wow, what a difference. And this was not N equals 1, of course. They showed many rats that were non-nurturing. So these groups had many rats. So what are the coincidence? What's the chance that all of them are behaving the same way? There's something there. There's definitely something there. And these ones that were highly nurtured, not anxious. So they said, okay, now let's dive into the genome. Let's look at their epigenome, their proteins. Let's look at their cell. Like what genes are upregulated, what proteins are upregulated, etc. So when in researching all of that, they said, you know what's really significant in both of these groups? The expression of GR. What is GR? GR is a glucocorticoid receptor. It's a receptor that binds to corticosterol. It binds to corticosterol. Corticosterol is the stress hormone. In rats and mice, it's called corticosterol. In humans, it's called cortisol. So when you get stressed, and suddenly you encounter something and that's stressful for you, your body releases cortisol. And that cortisol then goes and binds to the GR receptor. I'm going to refer to this as GR, short form. So it's a receptor, cortisol binds to it. What happens then? Then the GR receptor itself, believe it or not, is a transcription factor. It regulates transcription in the nucleus. So it actually goes to the nucleus, and then it activates several genes, and it inactivates other genes. So yes, another layer of knowledge that receptors can translocate to the nucleus. They can act in the nucleus, and the same molecule can be an activator for some genes and an inhibitor for other genes. So that's what GR does. Pretty big role, right? Now, when that happens, this is important. So cortisol is a good thing. You need cortisol. Don't put an X on cortisol and go, oh, the stress hormone is a bad thing. If you are running and you encounter a bear, you want to release cortisol. You want to react to that stress. If you're highly stressed for exam period, cortisol will help you because you know, sometimes you want that good stress. It gets you going. You react differently to things. So your organ, your heart's pumping faster, your organs are doing things that they need to handle the stress. So, but when that stress is gone, when the stimulus is gone, that cortisol has to be removed. And GR is the one that removes it. So the cortisol hanging out there is now going to cause havoc, is going to lead to stress and anxiety. And how does that do that? You don't have to worry about it. You can read up on it. We can't cover it in 50 minutes, but just understand that the excess cortisol, unless GR is present, it's just going to hang around and it's going to cause problems. And number one problem it causes is it leads to anxiety, anxious behavior. So they said, oh, okay, all right. So GR expression is low in these anxious rats and it's higher in these non-anxious rats. So I wonder if it has to do with mutation in the gene, if it has to do with mRNA processing, is it the protein turnover, is it a mutation? They tested for everything, but guess what they found? Everything was fine except for one thing, the methylation on the promoter. The promoter of NR3C1. NR3C1 is the gene that codes for GR. So I think I put NR3C1 on the final exam, so please write that down. Know that this is the gene that codes for GR. So NR3C1 has a promoter, like all genes. And this promoter is recognized by NGFIA, which is a transcription factor for this promoter. So when you want transcription of NR3C1, this transcription factor has to bind and activate this gene. But these anxious mice, when you look at their epigenomic sequence, you see hypermethylation of the NR3C1. What does that tell you? That NGFIA, which is a transcription factor, cannot bind, and now you get inhibition of this transcription. So you get less or little production of GR. In these mice did not show hypermethylation at the promoter, means that this transcription factor can bind, synthesize NR3C1, transcribe, translation, GR. Boom, expressed. So both of these sets of mice, these groups, right, all of these non-nurturing moms and all of the nurturing moms and their pups, they all had identical conditions. You can't be changing all the conditions, only have one variable, and that is grooming or not grooming. So they were identical. So you think these guys didn't go through some stress? They probably did, but they were able to handle the stress better. You can't avoid stress, live in a little bubble forever. You're going to be exposed to stress in your life. The question is just, how is your body handling the cortisol? So do you have the ability to handle it or you don't? And these mice just simply didn't. They don't even have GR. Cortisol's like, excuse me, I need to bind and I need to get to the nucleus. We need to do stuff. I need to activate a whole bunch of genes. Come on, let's go. And they're like, no, I don't have GR. No wonder they're anxious. Now, would this be convincing to you as a scientist to say, okay, well, you know what, I don't know about this. As you're experiencing the stimulus, the methylation pattern is changing. Maybe these mice were already not anxious. Maybe these were already anxious mice. Well, not really, because you have a whole bunch of them in the group. So the chances that all of them are experiencing the exact same thing and a coincidence is unlikely. But there's stats to prove those things. But there's still like scientists that are skeptical. I'm not sure. So, and they knew that. They're like, yeah, we need to keep proving this over and over again. So what they did, this is all the same paper. What they did was they did cross-fostering. Cross-fostering is when you switch the pups. So this rat here is a biological mom here, and this rat is a biological mom. So what they did was said, okay, let them give birth, and then let them groom and lick the way they do, whatever they're doing, highly nurtured, not nurtured. But halfway through, we're going to switch the pups. So now, these highly groomed ones are going to go to the non-nurturing and vice versa. So now let's see, do their methyl tags change? And guess what? They did. They did. So the ones that were anxious and had hypermethylation, they went to this mom, not their biological mom. not their biological mom, she's the biological mom. They went to the non-biological mom, but she was highly nurturing. And look at this, guys, the tags started coming off the NR3C1 promoter. And these ones, when they went here, what happened? The tags started going on to the NR3C1 promoter. Would that convince you guys? Would that be convincing? Or would you need more convincing? Who would need more convincing? Okay, I'm seeing some hands going, well, what if I said this was reproduced for the next 20 years? Okay, I got a nod, thank God. They also discovered that female pups, they raise their offspring the way they were raised. So when these pups, the females of these litter, when they became of age and they became pregnant, they were also non-nurturing. These pups here, the females of this litter, when they became older and became pregnant and had kids of their own, they were very highly nurturing. So yes, the argument is, and this actually does not translate directly to humans. There is evidence showing this, but it doesn't translate directly because there are, shown in human studies, that there are many parents that were raised a certain way. So whether it's good or bad. So if it's bad, they would say, you know what? It's just naturally in them, they raise their children the same way. Or they would say, I was, raised this way, I'm not going to put my kids through that. So they sometimes you see a drastic change in the parent. So it hasn't been proven. But and the theory is that, in humans, you go through life, like you have your baby in your teens, 20s, 30s, 40s, 50s. So you have time to change your epigenome, make decisions, and, plan out your life and what you want to do, what you don't want to do. Whereas in rats and mice, their lifespan is a year and a half. So they kind of just they're born, they're older in 6 months, they become pregnant, they have kids, and it's kind of like learned behavior. They haven't had enough time to have that epigenetic marking where they can now say, this is what I'm going to do, these are the decisions I'm going to make. So that's just one theory, but you guys can come up with your own theories, right, as to why this is happening. So another thing that kind of may seal the deal for you is when they took pups from this litter, which was highly groomed, so they did not have methylation of this promoter, what they did was they gave this group a drug that adds methyl tags. So it's saying, oh, you don't have any methylation, you have increased GR. Let's see what happens if we add methyl tags. In other words, we methylate this promoter. Do they become anxious? The same rat. Can the rat become anxious? It's almost like you're inducing anxiety. And guess what? Yeah, these rats become highly anxious. And you can really very well measure anxiety in animals. Like there's a way to measure whether an animal is anxious or not. And that's just solid data. So yes, they're highly anxious. So what does that tell you? It tells you that maternal care has a direct influence on your epigenome. Tags are literally being added and removed as the stimulus is occurring. And these tags can be permanent, they can be quite stable, they can have an impact, and it's, it can be passed on to the next generation. There is real data here. So this brings us to transgenerational epigenetic inheritance. Transgenerational epigenetic inheritance is the idea that epigenetic marks, such as DNA methylation and histone modifications, i.e. acetylation and deacetylation, can be acquired on the DNA of one generation and stably passed through the gametes to the next generation. And this definition, by the way, is on the NIH website. So what does stably passed mean? Stably passed means that when you have a methylation pattern in one cell, and that cell divides and divides, that methylation has to be stable in the daughter cells. You can't lose it. So we talked about that last class, where DNMT1 comes in, and it recognizes hemimethylated strands, and then it methylates it, so that for now the daughter cell maintains the methylation. So you're not going to pass anything on if your methylation is lost through division. You can lose methylation through division, We're not going to talk about that in this course called passive deletion of the methylation. But all you need to know for this course is that methylation is stable. And it gets passed on because to the next daughter cell because of that hemimethylated strand that's available, and DNMT1 comes and methylates it. And the only way you're going to pass on your epigenome to your offspring is if the change is in your germ cells. You're not going to pass on your heart cells. So changes that occur in your somatic cells, any somatic cell, heart, liver, lung, brain, etc., those are not passed on. The change has to be in your germline. That's now going to become your egg and your sperm. So that's really important to understand. So students often ask, okay, so what experiences are going to affect the germline epigenome, and what experiences do not affect the germline epigenome? So therefore, I may do this or I may not do that. It doesn't work like that. The research is not out there. Well, this experience will affect the germline, this experience will not. Maybe you guys will discover something later on, but right now it's just, we don't know. We can make inferences and say, well, this experience went on and this, but is there data out there to support that strongly? No. So experiences and environmental exposures can change the way your DNA works without changing the DNA itself, meaning the genomic sequence. So your epigenome can change, and this could be passed on to your offspring. Early life experiences exert long-term effects on gene expression, which can be inherited by the offspring. Can be, not always, it can be. So early life experiences, yes, we talked about those formative years 1 through 5, very, very strong marking. So a lot of the studies are showing that experiences that children had when they were very young, they can have long-term effects because really it's marking it in a different way. It's a little stronger. The marking is stronger, if that's an easier way to understand it. One thing that students always ask me, and it's a very common question I get, so I want to address it, just a couple of sentences. This will not be tested on the final. This is for your interest. Students often ask, okay, you guys know the epigenome is very critical for gene expression. If that's the case, then the epigenome plays a significant role during embryonic development. It has to. If epigenome is the one pulling the trigger, then obviously the epigenome has to be very prevalent during embryonic development. In fact, it is. It's a very strong epigenetic patterns, acetylation, deacetylation. These enzymes are working to their max during embryonic development because they're saying now differential expression is happening and the epigenome dictates that. So you're like, huh? That's weird. Okay, so if I got the epigenetic sequence from my mom and my dad into that totipotent zygote, then the zygote's not totipotent. How can it be totipotent then? If you have differentiation already happened, right? Which genes will be expressed, which ones will not? The epigenome has already said that in the egg and the sperm. That doesn't make sense. You know what happens in the totipotent zygote? Global demethylation. All your tags are off and they start from scratch. That's what happens. So now you're like, well, it has to, because it has to tell, make Hox genes, right? Make this thoracic region, make the legs. You have to give it instructions. It can't make instructions with an epigenome that's already established. No, global demethylation. So now you must be saying, so then how do we inherit the methylation pattern? 2 ways. Number 1, during global demethylation, and methylation is the main one they talk about because methylation is more stable, it's more stably passed on. During global demethylation, some of your tags that were given by your parents, they escape demethylation. They escape global demethylation. They're like, you're getting rid of all the tags, you're not going to get rid of me. So they escape it, number 1. Number 2, if they don't escape it, then guess what happens? They can have memory of which promoters were methylated and which ones were not, and they can put it back on. Isn't it wild how your cells can do that? Like, there's an RNA molecule there remembering how your methylation pattern looked in your parents' egg and sperm. So you don't inherit all the methyl tags from your parents and but you can inherit some of them. And this is how you inherit it, even though there's global demethylation occurring. So that was something that I think I hope answers a lot of your questions. In case you're confused, how do we actually inherit the epigenome if the zygote is totipotent? You guys know that, right? You can make the connection now. So they did similar studies in monkeys. And they still are doing research in monkeys, although there has been a lot of policies and restrictions around what can and cannot be done in these types of animals. PETA filed a lawsuit against NIH in chimpanzee research. So research with chimpanzees has had a really high, like a very big difference in how research is done in chimpanzees because of the fact that, PETA made some arguments and NIH made some arguments back. PETA did win a big one, and so they had to restrict and really change the policy on how research is done on chimpanzees, but they didn't win some of them. The long story short of it is the argument, and I'm not here to discuss the moral compass, I'm just here to give you facts. Chimpanzees became a big thing not because they're like humans, therefore it was more that chimpanzees are the few animals that people use in research that live 50 years. So if you induce trauma in a young chimpanzee at the age of 5, you want that chimpanzee to live 45 years with that trauma. So that becomes very unethical. NIH lost that one because there was a lot of substance there. For example, for rats and mice, the psychological trauma may be there, but it's not long-term because their lifespan is so little. But they're still doing research in monkeys. So what they did here was they randomized monkeys to 2 different groups. You can't do this in humans. You can't take humans from different families and just say, okay, you're going to go here, you're going to go here, and I'm going to do. It's preposterous. It doesn't happen. But apparently in monkeys, they can do this. So what they did was they said, okay, some monkeys are going to be raised by their biological mother and others with a surrogate. The surrogate is usually an inanimate object, not a living thing. So they have like a wire mesh and they put a blanket over top of it, and that's what the monkey holds on to for comfort in place of its mom. So this study did biological mother and surrogate, but please note down that there were studies where the non-biological mother was raising the monkey, and those results were similar when comparing mother to surrogate. The mother did not have to be the biological mother. Their epigenome were similar, whether it was mother, biological, or non-biological. So if you look at mom versus surrogate, did you guys think that there might be a difference in the epigenome of these 2 monkeys? Yeah, pretty sure, right? So what they noticed, and they kept the conditions identical. If the studies had them in cages, they were in cages. If the studies said, okay, cage time and then play time in the playground, like a zoo area, everything was consistent amongst both groups except for the mom. This monkey had access to mom. This monkey had access to a blanket. So what they noticed, guys, over time, is that monkeys in this group, they were highly aggressive towards humans and their peers. You would open the cage and they would be pretty much in attack mode. They were socially inept. They couldn't socialize. They had very odd social behavior in the playground. But you know what was really surprising, the most surprising result? The monkeys in the surrogate group, they were alcoholics. They craved alcohol. So they gave alcohol to both of these groups of monkeys. And they said, okay, you can either have it through a bottle or you can press a lever and the alcohol comes out. So they noticed that these monkeys, in the very first time, they tasted the alcohol, they loved it. They craved it, they wanted it. These monkeys here, they would smell it, repulsed, walk away. Wouldn't even lick it. This smell was repulsive enough that they just said, I don't know what this is, I'm not gonna drink this. I'm not gonna lick it, that's it. They would walk away. Right from the get-go, these monkeys, they wanted it. So what they noticed is they would say, okay, they would go in the playground, they would play, and then they would come back and they would press the lever and drink alcohol. It's almost like they would equate that as self-medication. You know how humans, if you look at it in the human world, it's self-medication. A lot of people self-medicate this way. They numb the pain with alcohol. And then that's how you eventually become alcoholic, because now you're dependent on it. So how do they say they're alcoholics? Well, eventually they would wake up in the morning, lever, drink alcohol, go out and play in the playground. That's a sign of alcoholism. They need the alcohol in the morning to function. So sad story. I don't know, you read these studies and you realize that life is a gray area, right? It's not black and white. I've become less judgmental over time, but of course you can't make excuses for people either that are doing horrible things and harming others, but everyone has a background, everyone has a reason, and some things are non-excusable and some things have a reason. So here you can see the methylome of these two monkeys, complete opposites. Red areas are of high methylation, green of low methylation. The monkeys reared by their biological mom or their non-biological mom, they had similar epigenetic pattern, whereas the monkeys with the surrogate had a very drastically different epigenome. So their epigenome is directly linked to their behavior and is also linked to the way they were raised. So the way you're raised, your environment, all the stimulus, how you perceive things, what you crave, what you want, it's all in your epigenome, and then your behavior is like that. And ultimately, guys, it becomes your destiny, right? So this slide is showing you how changes in the epigenome can lead to all of these things: risk-taking, depression, and these are studies proving these things. These are not just theories, these are actual studies. Schizophrenia is huge. They've shown a lot of evidence that people that are schizophrenic, they have changes, significant changes in their epigenome. And they actually now have epigenetic drugs for schizophrenia. They're called HDAC inhibitors, inhibit HDAC. Articles that talk about schizophrenia, they also talk about bipolar. So bipolar and schizophrenia, both linked to epigenetics, depression to epigenetics, all of these things. Drug addiction, interesting. They're able to treat drug addiction in rats and mice. They're having a difficult time doing it in humans because it's not translating very well. Drug addiction is a little bit more complicated. So one of the things to understand about drug addiction is the following. When these rats, or when humans, when they get addicted to a drug, cocaine, heroin, there's the addiction part, and then there's the memory part. The memory, and you don't realize that's what you're doing, right? That's how your brain works. The memory says, when I take cocaine, I feel this way, and I like it, so I want to do it. So that's the memory of the drug, the memory of the cocaine, memory of the heroin. So the mice remember that this drug, when I see it in this form, I like it. Oh yes, it's here, give it to me. I used to give cocaine to rats in my master's for another reason, and don't even go there. But when I would bring the cocaine out, they would just run to the cage. They were candida, yes, they're all crack addicts, right? And so I felt like this dealer. It was like such a bizarre feeling. And then they would just take the cocaine. But if you get rid of the memory that I have crack with me, and they look at me and they're like, I don't know who I don't know what she's got, I don't know what that is, they don't come, they don't want it. Because that memory is lost, that this is cocaine. That makes me feel good, I want it. So when that's lost, the addiction part is still there, believe it or not. But because the memory is gone, then the addiction. It's hard to get rid of addiction. Take a drug, I'm not an addict anymore. It's this tough. But the memory part's a little bit easier. So it's a step getting towards treatment. So it works in rats. They tried it in humans, flopped, doesn't work. We're more complex with drug addiction. These are all the articles that are out there, semi-recent articles, and these are not primary articles. These are systematic reviews. They're review articles. What does that tell you? This tells you that these are articles that are reviewing recent primary articles, recent research in that area. Look at all the journal articles that came out. Let's just summarize all this for you. So there's significant amount of data out there supporting the role of epigenetics in all of these things and how we need to come up with new drugs that are going to help us. Anxiety is a big one. Did you guys know that anxiety has exponentially increased since COVID-19? People are far more anxious since COVID-19. It has increased. So they did a bunch of studies on anxiety, understanding that anxiety is a big thing in society now. So a lot of rat and mouse models of anxiety. Look at all the success. Independent researchers showing success after success after success in epigenetic change, in epigenetic drugs in rats and mice. So they said, wow, we are ready for phase 1 trial. So valproate is a drug that they're using. It is an HDAC inhibitor. This is an older paper because this is one of the first papers to use epigenetic drugs in clinical trials. And then, of course, now you can read on studies that have stemmed from this. So epigenetics has a really big role in anxiety disorders. So you know what? If drugs in the past that are targeting the genome, they're targeting symptoms of anxiety, oh my God, symptoms, target the problem. Target the problem. If you target the problem, the symptoms will automatically go away. So if you target the symptom, the problem is still there. So these are approaches to target the actual problem. Get rid of HDAX, euchromatin state. We want transcription of this gene. That's the source. Some people have social anxiety, so they're looking at panic disorder, social anxiety. Some people are fine. These rats, they were fine in their cages with their family. But it's when you put them with other rats and other mice in a social environment, they have severe anxiety. So that's social anxiety. And humans have that too. Humans also have social anxiety. And so there are drugs that are coming out to help with this. So speaking of drugs and speaking of therapy, some of the biggest therapies in epigenetics is targeting cancer genes, targeting cancer. And I know a lot of you are saying, oh my gosh, you brought it back to cancer again. I know, I hope I didn't give you cancer fatigue, but it is what it is. I'm just telling you because this is where CRISPR-Cas9 is being used for cancer treatment. So here you have this tumor suppressor gene, for example, P53, the guardian of the genome, that's the target. So cancer cells inhibit the tumor suppressor gene. So therefore, now you get cancer. P53, or any tumor suppressor gene, has promoters. The promoters are hypermethylated. That's what cancer cells do. This dark stuff is what cancer has done. Cancer says, okay, let's go, DNMT, overexpress yourself, boom, 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 add methyl groups to the promoter. Now P53 can no longer be expressed. Ha, we got our tumorigenic state. So the treatment that the scientists came out with was DNA methylation inhibitor. They basically have a drug that inhibits DNMT, DNA methyltransferase. Do not transfer the methyl group to this promoter. So now the tumor suppressor gene can be expressed. Now, how accurate is this treatment? It's not very accurate, right? I mean, if you add a DNMT inhibitor, aren't you going to be inhibiting a lot of the DNMT across your body, like your cells, right? So it's not very specific. So what is specific is CRISPR-Cas9. CRISPR-Cas9 is very specific. How is it specific? Well, it has its guide RNA. The guide RNA guides it to the target. As you guys know, you've studied CRISPR-Cas9, and that sequence is very specific to that target. So now they can go and target the promoter of P53, not just random methylated promoters or random demethylated promoters. They're going to target specifically P53, for example, or specifically a gene of their interest. They can target it by saying, okay, I want to activate this gene, or I want to inactivate this gene. It depends on what they want to do and what the cancer type is and what gene is involved. So how do they do that? They use Cas9. D stands for dead. They could have come up with a better word, but dead means that it is catalytically inactive. It cannot cut DNA, double strand or single strand. So for the final exam, if I say Cas9, that's a double strand cut. That's the wild type. If I say Ncas9, nickase, that's a single cut for base editing. If I say Dcas9, there is no cut. So you might be saying, so what's Cas doing then? I don't understand. Why are you using a catalytically inactive Cas? You know why? Because they don't want Cas to go and make a genomic change. They want to make an epigenomic change. They want to say, leave the genomic sequence alone. Don't cut, because if it makes a double strand cut, now you're going to get NHEJ, HDR, you're going to get a change in the genomic sequence. That's not what you want to do. If you make a nick with Ncas, you're going to get something else. A base may be edited. You don't want to do any of that stuff. You want to leave the genomic sequence alone. You're just trying to methylate, demethylate, acetylate, deacetylate. So Cas cannot be catalytically active. So now you might be saying, so what's Cas doing then? Cas is doing two things. Number one, this guide RNA can't just go by itself to the target and bind to the target. You guys studied the natural immune system. Can the CRISPR RNA and tracer RNA do it by themselves? No, they have to couple with Cas, make that ribonucleic complex, and then that complex goes to the viral DNA, right? That's not how Cas works. Cas needs guide RNA, and guide RNA needs Cas. So together, Cas brings the guide RNA, and the guide RNA says, okay, I will stop whenever I get complementary base pairing. Boom, Cas, we're at the target. And Cas says, thank you, guide RNA, we're done. Their job is done. They're done. They're not going to do anything else. Now the real players, who are the key players here? DNMT, TET, HDAC, HATs. These are the things that you're going to couple now. Piggyback. This is going to be piggyback on DCAS. So do you remember in NCAS, what were the piggyback proteins? Cytidine deaminase and UGI. In DCAS, the piggyback are epigenomic enzymes. So in this example, it's the de novo DNMT, DNMT3A. What does it do? It adds methyl groups to a sequence that does not have any methylation. De novo, boom, boom, boom. And now, right, you have it. You have methylation of a gene that you want. You want methylation because, in this case, you want to inhibit the transcription of that gene. In this case, look what's happening. You want to demethylate. You guys know what enzyme demethylates? TET. TET now goes to the specific promoter, for example, the promoter of P53, which is methylated, and it removes the methyl groups. Boom, boom, boom. So now P53 can be activated because its promoter is no longer methylated. So now you have hypomethylation, transcription is on. Hypermethylation, for example, transcription was off. You can now control, guys, methylation in your cell. So this is being used to edit the epigenome very, very specifically. DNA methylation, or your methylome, can be sequenced. So they look at the entire methylome of your body, and they can determine your biological age. That's called your epigenetic clock. They can also look for biomarkers for early onset of diseases. So epigenetic clock is really cool. They can look at your epigenome and determine the age of all of your organs and give you an average of biological age. So what is the difference between your chronological versus biological? Chronological is by your calendar, the day and year you were born. How old exactly are you according to the calendar? But your biological age tells you how healthy you are. How old are you? If you're 20, are your organs 30? Start eating blueberries. So it's now going to give you a good idea of what your biological age is. So what do you want? What's your goal? At the very least, your chronological age should equal your biological age. But everybody wants your biological age to be less than your chronological age. And if your biological age is more, then that is called epigenetic age acceleration. Your biological age is accelerating more than your chronological age, and that's not a good thing. So I hope you guys all, have like a healthy diet and a healthy epigenome, and I wish you all the best. It was a pleasure to teach all of you. I wish you all the best in your future endeavors, and may all your dreams come true. I love you guys. Take care.